Hello everyone, let's talk about lecture 2 cardiovascular system part 2 heart. Heart is a transport system. Two side-by-side pumps. Right side receives oxygen, pour blood from tissues, pumps to lungs to get rid of CO2, pick up oxygen via pulmonary circuit. Left side receives oxygenated blood from lungs, pumps to body tissues via systemic circuit. Receiving chambers of heart, right atrium, which received blood returning from systemic circuit, and left atrium, which received blood returning from pulmonary circuit. Pumping chambers of a heart are right ventricle that pumps blood through pulmonary circuit and left ventricle that pumps blood through systemic circuit. Heart is approximately size of fist and located in mediastinum between second rib and fifth intercostal space, and also it is positioned on superior surface of diaphragm and two-thirds of heart to left of mid-sternal line and anterior to vertebral column, posterior to sternum. Base of heart leans toward right shoulders and apex points toward left hip. Coverings of a heart is pericardium, which is double-walled sac, and superior fibrous pericardium protects anchors to surrounding structures and prevents overfilling. Uh, Deep two-layered serous pericardium has parietal layer, lines internal surface of fibrous pericardium, and visceral layer called epicardium on external surface of heart. Two layers are separated by fluid-filled pericardial cavity to decrease the friction. Three layers of heart wall are epicardium, myocardium, endocardium. Epicardium is visceral layer of serous pericardium. Myocardium has a spiral bundles of contractile cardiac muscle cells. Cardiac skeletons are crisscrossing interlacing layer of connective tissue, and this anchors cardiac muscle fibers and supports great vessels and valves and limits spread of action potentials to specific paths. Heart valves ensure unidirectional blood flow through heart and open and close in response to pressure changes. Two AV valves, arterioventricular valves, prevent flow into atria when ventricles contract. Tricuspid valve, also called right AV valve, and mitral valve is called bicuspid valve or left AV valve, and codone tendon anchor cusped to papillary muscles and hold valve flaps in closed position. Two semilunar valves prevent backflow into ventricles when ventricles relax, and they open and close in response to pressure changes. Aortic semilunar valves and pulmonary semilunar valves are two types. As blood returns to atrium, pressure rises to open AV valves and fill the ventricles. As ventricles are filled, that pushes AV valves to close. Ventricles are discharging chambers. They have thicker walls than atria. Right ventricle pumps blood into pulmonary trunk, and left ventricle pumps blood into aortic aorta, largest artery in body. Equal volumes of blood pumped to pulmonary and systemic circuit. Pulmonary circuit is short, low pressure circulation. Systemic circuit is long, high friction circulation. Anatomy of ventricles reflects the difference. Left ventricle was about 3 times thicker than the right and pumps with a great pressure. Cardiac muscle cells striated, short, branched, interconnected, and one or two central nuclei. Connective tissue matrix endomysium connects to cardiac skeleton and contains numerous capillaries. T-tubules are wide, less numerous. Sarcoplasmic reticulum is simpler than in skeletal muscle, and numerous large mitochondria are present. Intercalated disc, which is junction between the cells, anchor cardiac cells. Desmosums prevent cells from separating during contraction. Gap junctions allow ions to pass from cell to cell and electrically couple adjacent cells that allow hearts to uh, behave as a single coordinated unit. There are three differences from skeletal muscle. Uh, about 1% of cells have autorhythmicity. Number two, do not need nervous system stimulation. Number three, can depolarize entire heart. All cardiomyocytes contract as a unit or none do. In addition, they have a long absolute refractory period, about 250 milliseconds, so this prevents tetanic contraction. There are also similarities with the skeletal muscle. Number one, depolarization opens few voltage-gated fast sodium channels and creating the reversal of membrane potential from minus 90 millivolts to plus 30 millivolts. And briefly, sodium channels close very quick. Depolarization waves also open slow calcium channels in sarcolemma and then um, sarcoplasmic reticulum to release its calcium, and it prolongs the depolarization phase, creating plateau. Action potential and contractile phase last much longer and that allow blood ejection from heart. Repolarization result of inactivation of calcium channels and opening of voltage-gated potassium channels create the calcium pumped back to SR and extracellularly. Heart rhythm can be altered by autonomic nervous system. Um, they are coordinated by functions of three. Number one, presence of gap junctions. Number two, intrinsic cardiac conduction system. Number three, network of non-contractile cells, which is autorhythmic cells. And they initiate and distribute impulses and coordinated depolarization and contraction of a heart. Pacemaker cells have unstable resting membrane potential, as you see in number one, due to opening of slow sodium channels continuously depolarize, and at threshold at minus 40 millivolts, calcium channels open, and this explosive calcium influx produces the rising phase of the action potential, and repolarization results from inactivation of calcium channels and opening of voltage-gated potassium channels. Let's look at the heart physiology. Sequence of excitation is shown. SA node is located on pacemaker of heart in right atrial wall and depolarized faster than the rest of myocardium and generates impulses of 75 times a minute. And AV node in inferior interarterial septum delays these impulses approximately about 0.1 second because fibers are smaller diameter and have a fewer gap junctions that allows atrial contraction prior to ventricular contraction. AB bundle or bundle of His is in superior interventricular system, and this is only electrical connection between atria and ventricles, and atria ventricles are not connected via gap junctions. And right and left bundle branches are two pathways in interventricular septum, and they carry impulses toward apex of the heart. Subendocardial conducting network is complete pathway through stir ventricular septum into apex and ventricular walls, and they are more elaborate on left side of heart. AV bundle and subendocardial conducting network, which is Purkinje fiber, depolarizes, and the ventricular contraction immediately follow from apex toward atria. Extrinsic innervation of heart, as shown here, heartbeat is modified by autonomic nervous system via cardiac centers in medulla oblongata. Sympathetic nervous system stimulate heart rate and force of contraction. Parasympathetic nervous system will inhibit heart rate. Cardio-acceleratory center stimulate sympathetic nervous system, which affects SA, AV nodes, heart muscle, and coronary arteries. Cardio-inhibitory center, which stimulate parasympathetic nervous system, that inhibits SA and AV nodes via vagus nerves. EKG or ECG is an electrocardiogram. They are composed of all action potentials generated by nodal and contractile cells at given time. There are three waves. P waves shows depolarization of SA nodes to atria. QRS complex shows ventricular depolarization and atrial repolarization. T wave shows ventricular repolarization. PR interval is beginning of atrial excitation to beginning of ventricular excitation. ST segment is depolarization of entire ventricular myocardium. QT interval is from beginning of ventricular depolarization through ventricular repolarization. Cardiac cycle shows blood flow through heart during one complete heartbeat. Atrial systole and diastole, followed by ventricular systole and diastole. Systole means contraction, diastole means relaxation. During this time, series of pressure and blood volume changes. There are two lambda heart sounds associated with closing of heart valves. First, there's AV valves close, beginning of systole, and second is SL valves close, beginning of ventricular diastole. There are three phases of cardiac cycle. Number one, ventricular filling. Please follow. There are three phases cardiac cycle. Number one, ventricular filling, which takes place in mid to late diastole. AV valves are open and pressure slow. About 80% of blood passively flows into ventricles, and atrial systole occurs, delivering remaining 20% of the blood, which gives end diastolic volume, EDV, which is the volume of blood in each ventricle at end of ventricular diastole. Number two, ventricular systole. Atria are relaxed, ventricles begin to contract. That will generate rising ventricular pressure and closing the AV valves. This gives isovolumetric contraction phase. That means all valves are closed. In ejection phase, ventricular pressure exceeds pressure in large arteries, forcing SL valves open, creating end systolic volume, ESV, which is the volume of blood remaining in each ventricle after systole. Number three, isovolumetric relaxation occurs early diastole. Ventricles relax, atria are relaxed and then filled by blood. Backflow of blood in aorta and pulmonary trunk closes SL valves that causes dicrotic notch, and ventricles totally closed chambers. When atrial pressure exceeds that in ventricles, AV valves open and cycle begins again at step 1. Cardiac output is the volume of blood pumped by each ventricle in 1 minute. cardiac output equals heart rate HR multiplied by stroke volume SV. HR equals number of beats per minute, SV equals volume of blood pumped out by one ventricle with each beat. Uh, Three main factors affect stroke volume. Number one, preload. Number two, contractibility. Number three, afterload. Preload is the degree of stretch of cardiac muscle cells before they contract. Most important factor stretching cardiac muscle is a venous return, which is the amount of blood returning to heart. And slow heartbeat and exercise increase the venous return, and increase the venous return stretches ventricles and increase contraction force. Contractibility number two is the contractile strength at given muscle length and independent of muscle stretch and EDV. And they are increased by sympathetic stimulation and increased calcium influx, and also by positive inotropic agents such as thyroxine, glucagon, epinephrine, digitalis, high extracellular calcium. And also they are decreased by negative inotropic agents such as acidosis, increased extracellular potassium, and calcium channel blockers. Uh, Heart rate can be modulated by autonomic nervous system, so sympathetic nervous system, which is activated by emotional and physical stresses, will increase the heart rate, and parasympathetic will do opposite effects. Let's look at this slide. How epinephrine increase the heart contractibility via a cyclic AMP second messenger system. As you see there, norepinephrine causes pacemaker cells to fire more rapidly and increase the contractibility. And epinephrine binds to beta-1 adrenergic receptor and increase the heart rate and increase the contractibility. And parasympathetic nervous system opposes sympathetic effects. As you see there, as we discussed, norepinephrine binds to the receptor. G protein coupled protein is now bound to GTP and then activated. This G protein now binds to adenylate cyclase, and then that enzyme will activate the ATP to cyclic AMP, and cyclic AMP will activate the inactive protein kinase to active protein kinase, which in turn do two things. It makes plasma membrane calcium channels to bring the calcium in, and also endoplasmic reticulum to release the calcium out, so it can bind to troponin, so that actin myosin interaction can be strong, so the cardiac muscle can contract with the force and velocity. And also sarcoplasmic reticulum actually has a calcium channels, which is also activated by active protein kinases, so that you can put the calcium back into sarcoplasmic reticulum, so that you have more stored to pump out. So this is how molecular mechanism signaling works for cardiac muscle contraction.